from the Radiation Research Society podcast. I am Manuela Bonanno from Columbia University, and very happy to have here our Marie Carie Award for 2017, Nils-Petter Rudqvist. And thank you for taking the time to do an interview with us. So Nils, please introduce yourself. Where are you from, where you work, and let's take it from there.
So my name is Nils-Petter Rudqvist. I originally came to the United States from Sweden in 2015. I work at Weil Cornell Medicine in New York, where I work. My mentor is Sandra Di Maria, and we do radiation therapy together with immunotherapy, and that's our research.
What's your background? Are you a biologist, a physicist?
My background is a little bit strange maybe for some, but so I have a background in physics, like traditional physics. So I've taken all these, subatomical courses, like, quantum physics and this kind of stuff. But I eventually wanted to maybe come closer to medicine, so I decided to take up radiation physics in the University of Gothenburg, and then I did my PhD in like more normal tissue complication after radionuclide therapy. And we did a lot of like gene expression work, and we did a lot of more biology. So I came closer to biology there, and now I basically shifted to a lot of immunology. So it's been going like from physics to a little bit radiation biology, and it's still radiation biology, I guess, to some extent, but it's more like radiation immunology, or whatever you want to call it.
So before we go through your work in the specifics and how you won this prestigious award, so you had a different background, a physics background, and then I can see that during your career you are very versatile. So from physics then you learned a little bit of biology, and now going more into immunology, and you look also at the clinic aspects. Mm-hmm. So what's your secret for being so versatile, and then at the end learning how to approach the problems in a different
way? I think I've always been driven by some people seem to have these grand plans, but I think in the end, for a lot of people, and also for me, I've been just driven by my curiosity. This seems to be very exciting, so I went that way, and that seemed to be interesting, so I went that way. So I have always wanted to understand things, and I have, when I have felt like this is so, exciting, fascinating, how does this work? And then I've been able, and I should thank all my mentors, and also that have believed in me. For example, Sandra, took me on board. Didn't know much. I didn't know the difference with CD4 T cells, CD8 T cells. I barely knew what a T cell was 1.5, 2 years ago, but I had something else to bring to the table. Maybe my physics background and my a little more bioinformatics or analytical background.
So we were saying, I was saying that Niels got this very prestigious award, the Marie Curie, and he submitted an abstract, and actually more than 400 abstracts were submitted, and you were selected for this award. Mm-hmm. So now, of course, I'm looking forward to learn more about your work and what you presented here.
Yes. First I would say I was very humble. I was very surprised to receive this award. I received an email from Dr. Limoli one evening in New York. Okay, that's strange. Why would he email me? And then I could read that I received this award. I was very happy, very surprised. So my research, the abstract, maybe I should say, Sandra Di Maria, was a kind of a pioneer in combining radiation therapy and immunotherapy. So she has a lot of different, she's trying to cover the area of understanding how we can improve the combination therapy in general. And my part of that is maybe trying to understand how radiation and the combination therapy can actually maybe shape the antigenicity. That is like how we can change the targets on the tumor cells by using radiation therapy.
So you look at the tumor response?
We do look at, yes, so I mean we look at the tumor response but we have done so much work now, so I mean we know, we have a very nice model, we know how it works, and so our immunotherapy treatment does not have an impact on tumor growth at all. Radiation has some impact on the tumor growth, but by combining radiation therapy together with immunotherapy, then as we use anti-CTLA-4, you can see...
Please remind us.
Yes, so anti-CTLA-4 is an antibody that blocks CTLA-4. So CTLA-4 is present on the T cells. And when the dendritic cells comes to present the antigens to the T cells in the tumor draining lymph nodes, the dendritic cells bind CTLA-4 on the tumor cells, and this creates a very big down-regulatory effect of the T cells. So basically, it's there to shut down the response, and it's very important. Otherwise, you might have a lot of autoimmune diseases and so on. But the problem is that the tumors seem to be able to, evade the tumor response because this happens. So in immunotherapy, this anti-CTLA-4 then is able to block the CTLA-4 receptor on T cells, and this enables the dendritic cells to instead bind these positive co-stimulatory receptors on the T cells. And that, together with presenting an antigen to the T cells, they are able to release and expand the T cells, and you have a huge activated T cell response that are able to have a systemic impact on the tumor burden. And this has been seen, in the immunotherapy trials, and they are approved now for treatment and so on. So, but the idea is then here to use radiation as a tool to improve this effect in maybe
because
patients.
Radiation might stimulate.
So radiation has radiation has a lot of impact on the tumor microenvironment, and you can increase the adjuvanticity. Basically, you have maybe an increased inflammatory response, which then stimulates the immunotherapy by itself. My research is more about the targets on the tumor cells. We're looking at what antigens are there and how can radiation actually expose the tumor antigens. So
what did you find?
So what we found was that, so we have looked at the T cells, so we do TCR sequencing, and by doing TCR sequencing we can actually measure what T cells are in the tumor, and since the T cells or the CD8 positive T cells are the ones that are responsible for the treatment effect, by looking at that repertoire, we can understand what targets they are maybe going for.
So let me go back just 1 second. You have a tumor model.
Yes.
And if you can just walk me through how your experimental setup,
Yes. Okay.
and then how you can then
I can briefly
sequence.
tell you.
Yeah.
So basically, we have a very metastatic mouse model of triple negative breast cancer. It's super aggressive. Within 30 days, if you leave them untreated, these mice will succumb. You can find 30 to 40, 50 metastatic nodules in the lungs that are visible to the eye. So it's heavily metastatic, and it's very hard to treat. It's completely insensitive to immunotherapy. And that's why we want to use this model, because immunotherapy does not work. So that's why we want to see how radiation can impact this. So we put the tumor cells, the 41 tumor cells that the tumor is called, in the flanks of the animals, and we wait for a couple of days. We treat them with radiation, 2 times of radiation, then we treat them with the antibody, that is immunotherapy, and then we kill the animals at day 22 where we see the treatment effect. And after that we isolate the DNA from these tumors, and we use that DNA to actually amplify the sequences that are specific for the T cells, and then we sequence this, and this information kind of gives us like a we can identify what T cells that are in the tumor by doing this kind of sequencing, because each T cell carries a kind of a barcode identifier that becomes visible by TCR sequencing.
So across mice, how much reproducibility you have?
The reproducibility, whatever, is is the treatment works like always, so that's very nice. But since the T cells are created within each mice, and it's created by this VDJ recombination process, that is randomly generating the T cell receptors that can identify the antigens. So we will find similar, tumor effects in each animal, but they will probably be rejected by different T cells. But this is all fine, because this is what is expected. One thing that was surprising for us was to find actually that we have treatment-related T cells that carry the same barcode identifier, even though you have a random process generating them. We could identify, for example, in the untreated group, specific T cells found only in this group that completely disappear where you have a successful treatment. And on the other hand, we can also identify an emerged cluster of T cells that are not found in the untreated groups or in any of the other like monotherapy groups that we use as a control. And this cluster of T cells can then actually be the ones that are responsible for the rejection of the tumor.
Okay, so what this brings, in terms of, this is more like two questions in one.
Yes.
So what's next for this particular project, and how do you think there will be practical applications?
Right. So so at Weil Cornell we have Sandra and Silvia Formenti, Sandra De Maria and Silvia Formenti. They are doing these clinical trials using radiation therapy and immunotherapy, and this kind of trials is going on other places as well. But we don't necessarily understand what patients to select. Like, who are the responders? So this is why we do the animal work, right? So the next step for us is we have the T cell response. So we have a big, actually almost it feels like a clinical trial by itself, but it's a preclinical trial where we have animals that we have pre-treatment tumor samples from and post-treatment tumor samples from. And by having that kind of information, we can actually more understand what T cells emerge, because the T cells come from the same animal, and since they are coming from the same animal, they have the same like clonotype, or how to say. So we try to figure out that, like what emerged in a single animal, and basically having each animal as a single patient, kind of, with pre and post treatment samples, and also we have blood from these animals. And that's one side of the story, but the other side of the story is that we're trying to identify the targets. So we're using a lot of the sequencing methods, like whole exome sequencing, RNA sequencing, and machine learning tools to actually predict the targets in the tumors. And this is important because the tumors, they have mutations, and from these mutations, new antigens that are completely novel to the mouse or to the patient are created, and these are believed to be one of the sources for the tumor-specific antigens that we then target with our therapy. So we're kind of working on both sides, like we're working on the targets, and we are working on the actual T cells that are responsible for the effect, and we're trying to put them together and show that radiation can actually expose tumor-specific antigens, and this is one of the sources of the responses we see in the clinic.
That's pretty exciting.
Yes.
And let me go back to what we were talking about earlier. So you are so versatile, and you can understand of many aspects of your project, and So what's your contribution to the project? So did you do the animal work, the sequencing? What?
So I always, my mindset is that I at least want to do all the wet lab methods at least once, because by doing that I can then learn and understand what happens later so much better. So I do animal work, I do the injections, I have people that are, Sandra has people that are much better in doing the injection, for example, of tumors, and so usually we collaborate. But I can do all these things, so I could be self-sustainable basically, but I prefer to collaborate so I can focus on the things that I'm best at. And I think my expertise is in more doing the analytical work, working with bioinformatic approaches and general analysis. And I guess that comes more from my physics background, being able to work with bigger, larger data sets. And so I think that's what it is.
And before we end, because you mentioned all these collaborators, and I happen to know many of the people working, Sandra, De Maria and Silvia Formenti. Would you like to acknowledge all the collaborators of this work?
So to begin, I would like to acknowledge, my Sandra, taking me on board, in this very cool project. I'm very thankful to her, and she probably helped me very much, like launching my career. Actually, I don't think there's a better place I could be at all. And Carsten Pilones, he was actually the one that initiated this project together with Sandra before I joined Weil Cornell, and I've been coming into the project and doing a lot of work in it, but it wouldn't have happened from the start without him. And so my project is also, I work together basically. I work a lot with another postdoc from France, Claire Lelier, and she's doing a lot of vaccination work, and she's more of an immunologist. So we work, so she covers the more immunological work, whereas I can maybe cover the more, the radiation work and understand that part in a better way. So I think Sandra's been able to put together a very nice team. And of course, Silvia Formenti, I've been very happy to be able to work with some of the patient data they have gathered over the year, and it's super exciting, and it's super hard to work with, because you have, the mouse word is easier, they are basically easier to work with, but the patients are where the effect is in, and that's where we need to go, and that's what we have to understand. And it's
it's challenging, but it's exciting.
It is very challenging. It is very exciting, and I'm very happy to be able to contribute to a therapy that I truly believe in. I really am.
So Nils, I really thank you very much for your time.
Of course, thank you.
I wish you lots of success, and hopefully next year you're going to tell us more about the project.
Yes, of course.
How it is going.
Yeah, absolutely.
Thank you.
Thank you very much. Thank you.